All right, so today's training, Oomycetes, aka water molds. And any of you who know me well know that I use the term water mold for all grower interactions. I rarely say Oomycete, and if I do, I always back it up with the word water mold. I'm okay if you, as agents, say water mold, that's okay. Very important, though, that you understand that we call them water mold for a reason. So if all of you recall, water molds require water to complete their life cycle, and they produce one spore type called zoospores, Z-O-O. Zoospores are the swimmers. They're flagellated and they swim in a film of water. So that is really a key, especially as we think about management. We clearly need water at some point in the life cycle of these pathogens. So water molds are primarily soil-borne. Pythium, Phytophthora, those are soil-borne pathogens, and they work their way either through root diseases or crown rots right there at the soil surface. Our terrestrial, or our above-ground Oomycete, is downy mildew, and there are several downy mildew genera. We're going to lump them all together, and we're going to generalize and say downy mildew. So your soil-bornes and then your downy mildews. We're going to kind of start out by thinking about them in two different groups. Again, Oomycetes need a film of water to complete their life cycle, but they can survive dry stages, and there are going to be some survival structures that can be produced if the fungus is allowed to continue to live long enough. It will go through a sexual reproduction stage and produce an overwintering or a survival structure spore or structure type. So we're going to think about those separately. Let's see, I'm going to keep myself on track because this can really get a little bit confusing. So let's talk about a little bit of terminology with your Oomycetes. They're all going to have some basic types. I was not able to get some live cultures for those today, so I am going to share my screen and we're going to look at some spore types. Let's see where I can get myself started. All right, bear with me here. Can you all, do I have to, are you seeing it? Okay, so this is a Phytophthora. Let me see, I'm going to go in order. Oh, I'm sorry. Nope, nope. Are you flipping through all of my, okay, apologies. Did you see the, here we go. This is downy, okay, this is downy mildew. This is a classic of downy mildew, so we'll start the round circles that you're seeing there. Those are sporangia, and I sent a little bit of vocabulary out in advance. Sporangia are capsules that are filled with zoospores. So look at, are we kind of going, my computer is not happy with all of these open windows, but you see here zoospores being released from this sporangium. So one distinguishing characteristics for those of you who like to play with microscopes is these branching sporangiophores. So the sporangia are held on these little branch sporangiophores. We like to call those antlers because they look like deer antlers, and that's pretty classic of downy mildews. Downy mildews are often confused with powdery mildews. Powdery mildews are true fungi. So if you're looking at downy mildew, so let's think about some of your outward symptoms. Leaf spotting, usually yellowing on upper sides of leaves that turns necrotic. On the undersides, we'll see mycelia, and that's the white body of the fungus. And within that mycelia, we'll start seeing branched conidiophores. And if you have a scope, you can see these antlers, and that's very distinctive. Powdery mildew is the opposite. You see powdery mildew on the top sides of leaves. It rarely causes any necrosis at all. And conidia are formed in chains of spores that go up. So just a little bit of nerd speak. So branched conidiophores are very classic. A lot of times when you put downy mildew on a slide or look at it this way, the sporangia are going to be knocked off. So you're going to see just those. All right, I'm going to close that. So that is downy mildew. Now let's go into, oh, I got some videos. Okay, so now let's go into, I'm going to skip myself over. So now let's go into some of the other pathogens. Oh, this is downy mildew as well. You see the branched conidiophores. You see the sporangia. They're a little bit, these aren't quite as round as the others. All right, so let's bump into Pythium. Pythium classically is going to show up as primarily mycelia. If you look at these hyphae, they are not heavily septated. Septations are the separations. So, and they're kind of granular. So anybody who's looking at that kind of granular in there, it's not a very clear white. That's one of the characteristics that we look for. All right, let me see. I have another Pythium here for you guys. Sorry. So here is Pythium again. Pythium creating the oospores, and oospores are the sexual stage. This slide has been stained. We're going to watch a video in a second. So you'll see those oospores. They're embedded within a root tissue. So a lot of times it's in the very fine roots, the very fibrous roots. That's what's going on. So these have been stained. They can look like bubbles. So kind of you have to train your eye and you have to zoom in and out and get that correct plane of focus. But your oospores are tiny. This is the sexual stage. These are the survival structures. This is what goes on underground. And this is why we tell you to remove infected plants right away, because these oospores can survive for many years in soil if not, if they're allowed to develop. So the contaminated soil, this is what we worry about. So oospores are the sexual stage. They are survival structures. They're usually a little bit darker and they have a thick wall. So they do resemble a bubble. They're just smaller. Okay, this is Pythium. Pythium here, we're back to asexual spores. Pythium, I'm sorry, Phytophthora, branched conidiophores, sporangiophores, I'm sorry, and the lemon-shaped sporangia. Sporangia are the capsule that holds zoospores. This is the asexual stage. All right, so asexual stage are the sporangia that hold zoospores, and they are the capsules. That is the repeating cycle. That's what we see going on when we have lots of water and ideal conditions, versus the oospores, which are the sexual stage that basically comes as a survival structure. So lemon-shaped sporangia, very classic of Phytophthora. And inside each of these sporangia are probably 30 to 60 zoospores. All right, I'm going through these pretty quickly. All right, now let's see something really nerdy. I'm going to try to, there we go. We're going to watch the release of zoospores from a sporangium. This is Phytophthora nicotiani. So this is, Kim, I can't see. Can you click play?
Sure.
I'm sorry, guys. So Phytophthora nicotiani is a tobacco Phytophthora. So you see the little zoospores swimming around. So they can swim for long amounts of time in free water. Free water meaning a very thin film of water. Doesn't take much. There they are. They have found a root of tobacco and they're going to infect. You see them finding that tobacco, being attracted to that tobacco. So this is really cool. So if you've got a scope, you could absolutely do this or watch this. So they're infecting primarily the tip of the root. And that is, as we start seeing symptoms, we're going to pull some roots out in a minute, we're going to see that it's the root tips that are infected first, and infection goes upward. So you're not going to see infection on sides of roots per se. Infection is always going to occur from the ends of the roots. So, okay, can you turn that one off? Oh, there was supposed to be music. We missed out on the music. All right. So I have to come around. You hit the escape button. All right, and we're going to watch one more video. This is zoospore release. So think about the life cycle as we go through, and this is something that we could not do live today, but look at those zoospores coming out of that sporangium. So in a film of water, there they go swimming again. So a film of water, and it doesn't take much. It can be high humidity, it could be dew, and there they go. So we can think about that as we talk about cultural practices and how cultural practices affect our management. So yeah, this is pretty cool and pretty fun. It's something that we will not be doing in the practicum in September, unfortunately, but this is always fun. And if anybody ever wants to see this in the lab, we can generate this for you just for fun. Okay, so if you want to watch any on your own, just Google, let me stop sharing my screen. Okay, technical difficulty. Oh, I just muted myself. Click. Maybe that. Click on your where's my, there it is. Sorry, guys. Somehow I'm having technical difficulties again. I'm sorry. All right. We have a frozen Zoom screen. Here we go. I'm going to stop sharing. There we go. I've survived yet again. So, so that was just a little bit about like what's going on and what we're looking for. We're looking for hyphae. We're looking for sporangia. Those are a little bit hard to see. Unfortunately, we can't look at those at the dyno light today. We're going to look just at symptoms. So some symptoms, let's go back through symptoms of oomycetes. Any questions, by the way? So I'm kind of looking at the chat box. All right, so let's start with downy mildew. Let's start with some symptoms. It's been very dry, so I could not even find fresh downy mildew, which is like a miracle because usually we have so much downy mildew and we could usually find it, especially in vineyards, but I could not find downy mildew. So these are pressed leaves. I'm going to show you these. They're pressed. They're not really full color, but I'm going to show you some examples of what downy mildew is going to look like. So this is a pressed leaf, and as you can, if I turn it, you see those yellow spots developing. This is going to be very similar to like a cucurbit downy mildew. You're going to see those yellow spots. They are going to turn necrotic in the centers as they get larger. The key with downy mildew is that it's really sensitive to UV. So you're going to see it, you see the mycelia or the actual signs of the fungus on the bottom side of the leaf. You're going to see them on inner leaves inside of the canopy, not on the outer edges, not in the very top like you would powdery mildew, because downy mildew does not like sun. So it's running away from the sun, if you will. Okay, you can see those yellow spots. I'm going to flip it over, and you're going to see coinciding with the yellow spots on the other side, you're going to see those white spots. And those white spots, I'm going to flip on the dino light in a second. You're going to see that mycelia. So here it is again. There's the yellow spot, and then the back side, there is the mycelia. So a little bit more spotted than your powdery mildew. There it is. Those are some pretty ones. So I'm going to flip on my dino light really quickly, and we're going to kind of look at this. Here's a little bit of necrosis going on. So almost a translucent yellow at first. So I'm going to flip. I'm going to share my screen to dino light. Let's see if it's going to cooperate today. That's what I'm getting dino light. Okay. I'm gonna lower my light. All right. So you'll see some of the mycelia there. Let it auto adjust. By the way, the light auto adjusts on your dino light, so you can kind of just let it rest for a second. So here you see, now this again has been pressed. We've knocked it around, so it's had kind of a really hard life. This one Kim uses for a master gardener, so it's been handled quite a bit. But you see that mycelia, it's crawling around, it's skating around on the surface of that leaf. Let me see if I can go a little bit further. Is it gonna do it? Remember, we have that second plane of vision with your dino light if you just keep turning. So there is the mycelia. So if we would put this mycelia on a slide, we could see the branching and we could see the antlers. So there we are. So downy mildew, usually pretty fluffy. Not all downy mildew is this fluffy. Look in, let's see if I can, where's my pointer? So here in this area, oh, well, see here in this area, you see all of the round things. So we have some clusters of mycelia, and we also have some sporangia going on there. So that is what downy mildew pretty much looks like. Any questions? Get back in my focus. So you see the fuzziness. I don't know how to make the red arrow go away now. So there's downy, there it is in some small spots. Here is the necrosis and yellowing on the upper side of the leaf, and then coinciding. There we go. That would be pretty fluffy growth. Now, like your basil downy mildew is not as fluffy. So it takes a little bit longer. You have to do a little bit of incubating sometimes. Any questions on downy mildew? All right, I'm going to move on. I'm going to unshare the screen. All right, so, okay, last call for questions. All right, so let's look at another type of pathogen. Let's look at Phytophthora root rot. Everybody is familiar with Phytophthora root rot. So Phytophoras primarily are your root rots, crown rots. So what happens when you have something like a root system that is covering an entire plant? The entire plant depends on the root system and then on that crown. So when you start getting loss of roots and you start losing that that phloem tissue, then you have lost the pipelines of vascular tissue to the plant. You're going to get wilting first, you're going to get collapse of the upper plant parts. If water and nutrients can't make their way up and down the plant, then you're going to lose the turgidity first. You're going to see some wilting in the early stages, especially on hot days. So you're slowly going to lose vigor. You get a really hot week, like a couple of weeks ago, you're going to see starting quick collapse in plants. So that's pretty typical of the symptoms. So wilt in particular. Once you see plants wilting, backtracking and so what can be happening and looking at symptomatic plants. So I'm going to start with these are cabbages. This is a Phytophthora crown rot. This is going to be a dry rot. Typically, Phytophoras have very wet symptoms. So they're water molds, remember, they need water. So it's going to be pretty wet symptoms. I got these from, let's see, I got these from the diagnostic lab, and they've already been hacked on. But I want you to take a look at this crown rot, and it pretty much, is it going to focus? It pretty much look like a, there we go, kind of like a canker. So you can see the canker margins. Let me see, we might have to switch to Dynolite again. We see this canker margin here. There's my pen. So you see anything down here, and then you see this margin here. So whatever this is, it started down low. Look at this really bad root system. Started down low, and it's working its way up. So this is an indication that this is something that's soil-borne. It started from the bottom, and it's working its way up. See what we can adjust here. So I am going to use a scalpel. You can use any kind of blade tool. So what we are looking for is discoloration at that canker margin. Let's see if I can do this backhanded. So there is clearly the canker margin. You'll see it, try to roll it here. You'll see the movement here. This is an older sample. Phytophthora is typically red. We usually call this the ring of fire because it usually comes up in flames or something resembling flames. There we go. There we go. So looking at this, we know there's something wrong here. Okay, we know that there is a crown issue. Now, if you were a diagnostician, you would be targeting the margin of this canker, and this is where you would sporulate. This one is not sporulating on its own. This is something that we would sporulate in the diagnostic lab. So as you're submitting samples, and something as small as a vegetable, or even your row crop plants early in the season, you can submit entire plants. When it's a woody plant, it's a little bit harder. So here you have your canker margin, and I'm calling it a canker, but that's probably an incorrect term. So this is a Phytophthora. This one is dry. This is a rare. It's rare that Phytophthora is a dry rot. It's usually, oops, sorry. It's usually a wet rot, and Phytophthora typically stinks. For me, that's always an indication when you have a stinky rot. But this is an oddball. This one is dry. But I do want you to look at some of these canker margins. And hopefully in September when we do the practicum, we'll be able to get our hands on some of these and let you whack away and find some of these canker margins, not just in the Oomycetes, but in some of the fungal cankers as well, fungal and bacterial. Any questions there? Okay, so I'm going to unshare the screen. All right, so again, I'm going to show you that I had two cabbages. They have Phytophthora root and crown rot. You'll see the limited root system here, very typical of what starts out and the root system. and works its way up, worked its way up the crown, we'll see that girdling. And with that girdling, we have disrupted the flow of water and nutrients up to the upper plant parts. So we started seeing browning. It's kind of hard for cabbage to wilt, right? But what would be wilting if you were a cabbage? So you clearly are seeing the dull color. Then you see wilt, and you could easily see collapse on a hot day. So there it is. It's a Phytophthora dry rot. Questions? Okay, let's look at another Phytophthora. This one is tobacco. This is one that you all probably are much more familiar with than I am. It's falling apart. So this is a tobacco plant that has black shank. So we don't see an effect on the root system. This one is going right to the crowns. So browning and collapsing. Let me see what I can find for you here. I am going to cut this guy apart just so I can work with it. And I am making a huge mess in my office today. We have a lot of dirty samples today. So I am going to see if you can see. No, you can't see that canker margin. I'm going to have to flip back to the Dino Light. You can see the kind of the girdling happening here. Maybe a little bit harder. Here we are. Here we go. It's a little bit darker. This is the canker margin. Now, of course, this guy is dirty. So I've got some water. Get a paper towel. See if I can attempt this without making a muddy mess in here. And there it is. So if you would have rinsed this out in the hose, you could have there we go. You can see that very distinct, the girdling that's happening. This is sunken. If I turn it a little bit, you can see the sunken areas. And this is very typical of black shank. So same thing. We've got a Phytophthora, we've got a pathogen. Let's see if I can get rid of this arrow. Can you hit the delete button, please? Yay. Okay, I'm sorry. We still had that red arrow going on there. Okay, so here I am again with my scalpel. You can use anything that's sharp. And the key is to keep a very light, that's too deep, to keep a very light slice in there. And this is your classic black shank. There's the margin of that canker. So that ring of fire is moving its way up. Oh. And you have to stay thin. So see, there we go. So this is mushy and wet. So as you can see, I'm just kind of like mushing it up here. So this is what would be sporulated in the diagnostic lab. So sporangia developing in this area. If you sporulate this guy, sometimes if it's really rainy, you're going to get those sporangia, they're going to be visible. So that is, remember the sporangia is the asexual stage. The sporangia are those capsules that hold the swimming zoospores. So think about the weather. Think about when black shank will establish. And once it is established, it can kind of just hang out in the mycelial stage until we get some wet conditions. So there it is, guys. Black shank of tobacco. Very clear, black, of course, hence the name. Not that disease names always coincide with symptoms, but this one is indeed black. And again, it has come into the crown, and our roots look pretty darn good. So this is not a root rotter. This is a crown rot. So back again. Any questions there? All right. All right. Switching gears. I'm gonna unshare my screen again. So we're moving faster than anticipated. So we looked at two different Phytophthora, a dry rot, the rare one. We looked at a wet crown rot, which is black shank of tobacco. Coming in, that is a crown rot that does not affect the roots. Root rots, usually Phytophthora likes warmer weather. On the flip side, Pythium likes cooler weather. So we usually start seeing Pythiums in the springtime. Phytophthora will usually come in a little bit later in the season. A couple of other things about Phytophthora, it can come in a lot of herbaceous plants, especially greenhouse conditions, and especially it can come in early. We move it around, we move soil around a lot of times, and once again, it can establish in a field. Once those sexual spores create the survival structures, then you've got a long-term issue. Whereas if you're in a greenhouse, then you can sanitize that greenhouse, not keep that crop long enough to let it get to that stage. So that's usually like, say, an ornamentals greenhouse, spring or summer hanging baskets, for example, that being brought in probably either by maybe a mulch pile outside, something that built up, or coming in with plugs or liners. So always, and we don't talk about management in this series, but backtracking, where did this come from? If these are soil-borne pathogens, it's not blowing in. Where is it coming from? From shoes, from soil, from dirty hoses, anywhere that soil can be transferred. But what is the original source? Sometimes that original source is a natural source of inoculum, but a lot of times we're bringing it in some other way. And then once it builds up, then we're kind of stuck with a field or a high tunnel, some bed that has a long-term case of Phytophthora or Pythium, for instance. So any questions on Phytophthora? All right, I'm going to switch gears. I have an ornamental this time, just as an example of Pythium root rot. So we're switching to Pythium. Pythium is typically a cool season. Oh, I'm not very clear. I'm a little bit blurred. So this is Pythium root rot in chrysanthemum. So a lot of mum production here in the state. We see more and more mum production and pretty large areas. Some of our tobacco growers even growing mums for commercial production. Not just our ornamentals people, but I even have a few orchard growers who are producing their own mums. Commercial mums are grown in containers, but they're grown outside in on black plastic or black cloth. They call them beds, but they're not beds. That is just it looks like a nursery production area. Mums are typically planted at the very end of May or into the beginning of June. And then they are grown to flower right at that September when we start wanting to see fall crops. So this is one, Pythium often comes in with the stalk, or it's coming from the ground. So the ground cloth that's coming in under them, usually once infected, and then it's porous. So the mum pots are sitting, basically they're touching the soil. They've got that fiber underneath them, but it's coming back up. So if you think about the movement, if you're moving this disease in water, just like any of the others, in this case, it's penetrating down with the water and it's contaminating the soil underneath the cloth and it's overwintering there. And in this next summer, when you're watering and you've got these puddles, then that Pythium is being transferred back up into the pots. And Pythium loves mums, amongst other things. It loves fibrous root systems, very soft root systems. Pythium prefers, again, cool season conditions, but Pythium coming in June is not unlikely. So once infection occurs, there it is. Cloudy, rainy out there today, that's perfect conditions. It cools off just enough for that Pythium to become active again. So, and I'm generalizing, I'm telling you about this mum, but think about it in the larger context. How does Pythium work in a tobacco float bed? How does Pythium work in, say, a new soybean planting in cool season if you plant early, or if you have a wet spring. So think about it in that entire context. So let's forget, if you don't work with ornamentals, let's forget that this is an ornamental at all. So one thing about Pythium is, look at the root system here. You've got a root system. Pythium is a root nibbler. It likes to come in at the ends of roots, and it just kind of hangs out there at the ends of roots, which that's the feeder root. So even though you have a root system here, you've got infection at the root tips. So one of the characteristics of Pythium is blackened root tips and what we call sloughing off. I am going to switch again, and I'm going to show you what sloughing off means. It means the cortex of the root has sloughed off and we're left with just the steel. I'm going to share my screen again. This is something you can absolutely do in your office. Let me see if I can clear this up a little bit. I think I need a colored background. Where's my black paper? Oh no. Let me see. I'll try a paper towel. So I've got so much dirt down on the bottom of this thing, it's hard to see. So there you see the cortex broke off right here, and then there is the steel, which is the center of the root. I'm going to go down for you. Put this into focus, and there it is again. So there's where the cortex broke off, and you've got the steel that's left. We call that a rat tail. So what I'm doing is, I don't know if you saw earlier, I've got a bowl of water on my desk. So I'm just going to dip this root in there and see if I can clean it up just a little bit so you can see a little bit better, and we'll look at more of these. Not really, huh? There it is again. So you see the rat tail symptom, which is, this is always awkward to look at it on the screen and not straight down. Okay, there we go. So we have our root that looks pretty good, and then we have that sloughing off. So I'm going to switch, and I'm going to show you an entire sample. So I have another much more severely damaged sample here. Is that clear to everybody? If it's not clear, let me know. So you see there's still soil intact. And you see right where we see we've left the soil here, then we have that sloughed off, and then there's that rat tail. There is that just that steel hanging out. And if you really don't know the difference, get a fresh root. You're not going to take off that cortex. That cortex is attached there very tightly. So an important skill to learn when you start seeing Pythium, especially severe. Once you start seeing major wilt, you're going to see this sloughing off of roots by Pythium. Phytophthora will eat it away. Pythium is going to nibble at the ends, and once it gets high enough, that cortex is just kind of just slimy and wet. All right, I'm going to unshare. All right, so this is that second sample that I had. See how severely damaged it is, and then you don't have a single decent root in there. Look at that. Let's see all the rat tails in there. Pretty gross. Now, when you have a sample like this, here's my bowl of water. This is where the mess really comes in. So this is just any bowl of water. You're just rinsing the roots off because you want to get the soil off so you can see. The last thing you want to do is do this in your sink. In our diagnostic labs and in our dirty labs, we have a soil trap right where the S-trap under your sink happens. there's a reservoir so that those sediments can drop down, and then we can empty that on a regular basis. You don't have that in a typical faucet sink situation, so you're absolutely never going to wash a lot of this stuff down the sink. Just get some kind of bowl of water, a bucket, outside in the garden hose. It doesn't matter how you do it, but you're just going to rinse the soil off of this sample. And the more you do that, the more you're dislodging, in this case, media. And then you can come back and you can separate those roots. I like to cut them apart just because it's cleaner to cut them apart. In the diagnostic lab, we use a Petri dish. We use a Petri dish, but then we can come in, we can rinse them a little bit better. This one getting pretty dirty, so I would probably cheat and go to the faucet at that point. And then they come into, then they come under the dyno light this way. So so coming under the dyno light this way, and you can tease these roots apart and see that sloughing off. You can photograph these however you choose to do so. So this is a very kind of classic diagnostic of the Pythium, of a severe Pythium on an herbaceous plant. Any questions there? So we have a little remote for my camera. All right. So Oomycetes. Let's backtrack. Let's give a little bit of a review. Oomycetes come as soil-borne pathogens and terrestrial. Terrestrial Oomycetes are your downy mildews in particular. Downy mildew sets in when it's rainy or when it's wet, really humid, sometimes irrigated areas or under a shade tree, in a holler, at the bottom of a hill, wherever you're going to see enough moisture that you've got a film of water. And film of water can come as dew, and it could come as different high humidity type situations. So downy mildew, terrestrial. Your soil-borne downy mildew, your soil-borne Oomycetes, or your Pythiums and Phytophthora in particular. Those are the ones that we deal with on a regular basis. So let's backtrack. Pythium likes cool weather. Phytophthora likes warmer weather. Pythium prefers herbaceous plants. Phytophthora will affect both herbaceous and woody plants. Both of them come in at root systems. They can come in at crowns, especially if there's any kind of damage. They really like root tips because that's just really easy to infect. So they don't have to work real hard to get into those root tips. But they have to work hard, and they're not capable of working so hard to get into some of the woodier root areas. So we always talk about damage. We always talk about wounding. This is one of those cases. Pythium will be our root nibbler, so it'll start nibbling at the end of the root. It never completely destroys roots, but it'll inch its way up, and over time you're going to get that sloughing off of the cortex and be left with the steel. Phytophthora, on the other end, will destroy a root. So it'll start from the tip, but it'll destroy the roots, and then it can get into the crown or into even the trunk of a tree. And we call it when it moves up the ring of fire. So it'll make like flames. So you'll see it come in, and you'll treat that like a canker when you're sampling and when you're examining it. No questions there? Okay. So it is, it's just 10:09. So I want to show you something today that is a little bit kind of off in a contrast to our Oomycete root rot. I want to show you a fungal root rot. I did not have this one in our fungi day, so I want to show that to you. My office is a mess. On our last session, I'm going to pan out and I'm going to show you this office. We have huge stacks of boxes for our September training. We've already purchased those supplies and all those goodies, in addition to lots and lots of dead plants and stacks of papers that I'm supposed to be dealing with. So almost comical. All right, so this was a holly. Okay, it's a nursery container. I took a photograph of it because I had to cut it back. It barely fit into my cart. It was about 4 feet tall. This is a diagnostic sample, so if it belongs to you, you might recognize it. So the entire holly was dead. It just completely died back, the entire thing. I looked for cankers, or the diagnostic lab did first, looking for cankers. There were no cankers. There was no flagging. There was nothing indicating cankers. So in our diagnostic protocol, we looked at a common denominator, which would have been the crown, this multi-trunked. Looking at the crown looked really good. So then the suspect was roots. Because it's a holly, we are suspecting automatically black root rot, which is the Leviopsis. That's a fungal pathogen. In this case, okay. Going back to the okay, DJ, I'm going to come back to that in 1 second. So I absolutely want to cover that. So we are we are suspecting that this is probably black root rot, which is the Leviopsis, and it's a holly, so we're almost guaranteed. As an aside, for those of you who work with ornamentals, my counterpart at UT, Tennessee, Alan Wyndham, who works in Nashville, did a project a few years ago, 2 or 3 years ago, and he sampled 100 holly plants in retail nurseries in Nashville, and 100% of them had black root rot. So that is just kind of a little tip that you kind of know what's going on, and you don't have to grasp for straws or look for a needle in a haystack. You can zone in on what you think this is. So I'm going to take this guy out of the pot, and I'm going to walk you through how you would look at something woody this way. And I want to show you this black root rot. Now, we have a question on Phytophthora root rot and just how do you sample roots on a tree. So a mature tree or something large, you just can't. We are going to, so usually if it sets in onto a large tree or a large woody plant, it's coming in early in that plant's life. It rarely comes in later. And so with that time, it's going to start moving its way up into the trunk, into the crown. So look for right at the soil edge, look for sunken bark or something that might not be it won't be a canker coming from the outside. It'll be underneath the bark. That's where the Phytophthora is going to work its way up into those flames. And so the bark might be soft or like not quite connected. So if you kind of press it with your thumb, it'll kind of be wiggly in there. Also, it'll be sunken. So any infections are going to result in dead cells, and the cell's not growing there anymore. Whereas the healthy cells around it and surrounding areas are going to continue to grow. And that results in kind of a concave or a flattened type part of the tree. If you suspect that, and if the bark is loose, you can take a pocket knife or even your thumb and pop the bark off and see if you see that discoloration. From there, you would sample the margin of that discoloration, and that would be submitted to the Plant Disease Diagnostic Lab. Our Kim is working on a canker sampling publication, just to kind of show how you sample for cankers. It's not quite as cut and dry as we kind of tell you, so we are going to, it's basically going to be a photo how-to so that you can look through it and kind of get some ideas. So yeah, Phytophthora or any root rot, any crown rot is very hard on a woody tree, especially if it's large. This is really as large as I want to go. This is a heavy pot. This is probably a 7-gallon. I don't know how it's sized exactly. It's about a 7-gallon, and it weighs a lot. It weighs about 30 plus pounds. And so hauling these kinds of things around are really tough. And so we can flip this one out. We can manipulate this one. If it was in the landscape, we could dig it up and we could manipulate it. But anything bigger than this is just not easy. If a plant is not completely dead, very often your homeowners in particular are not going to want you to dig it up. They're not going to want you to sample it. They're not going to want you to drill it or saw off a section of the root system. And that's really a challenge. If we suspect a root rot or a crown rot, we've got to have a sample. And we've had situations in the last few years where we just couldn't move any further because that stakeholder just did not want to sacrifice the plant. And I wish there was another way, but it's not. There's just no other way. We have to have a sample. All right. So, oh, if you see the green happening at the base of this plant, the plant wasn't completely dead. There was enough green tissue here. So I've had this plant, been hanging on to it actually since our fungal workshop, which was several weeks ago, I think middle of June. So I ran out of time. I wasn't able to do this one. So I wrapped the root ball in a garbage bag and I've hung on to it. I kept it watered, and obviously there is enough root tissue to reinvigorate a little bit of growth. So it's never going to be okay, but that's the green that you're seeing on this plant. So I'm going to take this guy out of the pot. He was out of the pot yesterday. This is why my office is a disaster, because we do these kinds of things on a regular basis. All right, so here's a big whopper root ball. When I look at this root ball, and here we go, right away, we see there's soil in here. It's not just media. And in container grown plants, we cannot use soil. So this tells us that there was a problem from the nursery aspect. Also, if you look in here, now we have worked in here. You can't see. If you look inside, I'm going to flip my dyno light. You see sand. See, it's kind of white in here, and it's sand and gravel in here. So apparently, the smaller liner was in some kind of sand or gravel. I don't know. I don't know what the heck is going on here. Maybe this was a smaller balled and burlap plant, and then they just kind of backfilled with some media, but this is clay. This tells you right here that there are bad cultural practices altogether. But the most important thing we are looking at here is look at the root color. All right, remember our roots are supposed to be nice and white and turgid, and these are black. Woody roots don't flatten quite as easy. The feeder roots will. You don't see a lot of feeder roots, and you don't see really any white roots, or not many white roots. So I am going to switch to dyno light, and I want to see if we can focus on some of this. All right, bear with me, everybody. What does that look like on your end, Kim? So we see some, oh, it's very dark, apologies. Let me come closer. So we see a lot of black roots. A lot of black roots. So we see some white. Ooh, jackpot. Okay, so we see some white. We see a lot of black, a lot of gray, a lot of discoloration. The white ones are showing up, but look how many black roots are back there. Okay. Let me see. Oh, I'm touching the photo. I'm touching the photo button on this dyno light. I always do. So look at the blackness in these roots. And that is suspect. So what's going on with these roots? We question, is this Phytophthora or is this black root rot? And based on what we know, we're going to suspect black root rot right away. So what I have done is I have used a pair of pruners, a pruning saw, and razor blades, and lots of combinations of things to hack away at this sample. And I am going to show you what I ended up with. All right. So black tips on these roots or black roots. So we've seen some feeder roots, a lot of the feeder roots being intact. We see that they're black. And if we backtrack in, we have some white. Okay. Let me flip this guy over. And there we see some black lesions. And one thing about black root rot is the lesion. So it's coming in on ends of feeder roots and it makes its way back. So black root rot is not the destruction per se of all of the root tips, but it follows its way back and there it stands. So black root rot, very well known for its ability to infect in middles of roots and cause lesions in middles of roots. Here, they're fluffy here. So I'm going to come down a little bit with my dino light. So examining these types of things. So if we suspect black root rot, and we suspect black root rot simply because we have done our homework, we've used reference books, and we know that that is the first thing we would check for with a holly. All right, I'm having problems with all the dirt on my desk driving around. See, so we've got some lesions, some black lesions here, and this is just an example off of the one that I sampled. So let me see what I can do. See if I'm gonna. So see that lesion in there. I cut it off. So we see these lesions happening, and that is one distinguishing characteristic. So we know we have the equivalent of a canker in the middle of a root. And that one was too deep as well. So being able to lightly scrape and to do it in a screen is a lot harder. When you're working at your desk, and when we work at this for the practicum, it'll be much easier. So we see these lesions, we see these equivalent of cankers in the middle of the root. This is not a root tip, it's not a root feeder like we see in our Pythium and Phytophthorus. It's in the middle. So this is a fungal disease. This is a fungal root rot. You'll see the peeling of the cortex here. So anyway, I always think this one is fun. Let's see if we can see any structures on this thing with our dino light. I cut some of this off just so I could have it lay still and flat. And go all the way down. Pretty dirty. Let's see. It's hard sometimes to tell soil from your go the other way. Let's see if I can go down that extra plane. Remember, again, when you have a sample, you can always go down an extra plane through with your dino light. If you just keep cranking, it'll give it something else. All right, I am on my paper towel. So you see the fungus here. And if we had a little bit more magnification, we could see the chlamydospores, which are which is a survival structure that lets this pathogen hang out in landscape beds and in soils for extended periods of time. Any questions? Go up just a touch so I can move my sample around. No? All right, I've maxed out the capability of my dino light here. And it is so we see fungal some fungal structures, not real clear. We see some fungal structures. So this is beyond the rot. So this is an old lesion. All right. Any questions? Unshare. So we've kind of really come to the end. I wanted to just briefly cover a little bit more about roots, even fungal type root disease, while we were covering oomycetes. But oomycetes, aka water molds, different from your fungal spots. Okay, it's the moment of truth. Any questions? I'm always open to questions anytime. Okay. Did you answer this one? Yes, I did. Thank you. Okay, I'm trying to share my screen and it looks like we're locked up. Let me stop that share. All right, let me try again. Kim is not happy with us today. We were trying to share the link to the survey. Oh, there it is. Okay. We are in luck. I am going to share my screen again. Remember that the website is the training website is always up, plantdiseasetraining.ca.uky.edu. If you click on webinars, then you can go into all of the webinars in reverse order. So this webinar number 5 is going to show up at the top. Scrolling down, that's where all of the resources are. The quiz, here is the link for the quiz. Kim is putting it in the chat box now, so if you want to click it live. Okay, one sec. So very important is that if you have not taken quizzes 1 through 3 at this point, we are not going back. Quiz 4 is also closed. We closed it at 9:15, just when this one started. So we have a couple of people who have asked for extensions, but we knew those in advance. So this is webinar 5, oomycetes, aka water mold. So this is your link. And I'm going to unshare my screen. And Kim has put the link in the question box. So we have a question. What do we tell the what? The homeowner? What? I can't read that. They can ever do anything preventative. Oh, for black root rot. Okay, what do we tell the homeowner about black root rot? With black root rot, just not planting susceptible plants there anymore. It's unlikely that they're going to be planting holly for 5 or 6 years. At least 5 or 6 years, possibly more. So I would help them go through a list of plants that are not susceptible to black root rot. A lot of your annuals are, for instance, pansies, petunias. We see a lot of black root rot in pansies. So to go through and help them through that. But holly is just really not going to be an option. One thing about a lot of our pathogens is that they are kind of host specific. Some are very host specific, some are generally host specific. So if you have a host specific pathogen, then we can use different hosts for replacement. And we see that in every circumstance. We see that in row crops, we see that in ornamentals, we see that in vegetables, even in fruit. So we're always gonna work with them. And that's why the biology is so important. We don't like to talk about the biology, but darn it, we need to. Because knowing how these pathogens live, how they overwinter, how they infect, where and when they infect, we're able to manipulate those systems and use cultural practices to get around even the most severe of pathogens. Any other question? Did that answer your question, Kathy? Okay, good deal. Anything else? It is 10:29, so we did not finish early, did we? I'm reading another question. Can Phytophthora occur in mature trees after injury? DJ, it can, it's less likely. Usually it's out there, it's kind of maybe infected, just not having the conditions for it to really explode. So let me backtrack. Remember that infection can occur days, weeks, months, even years before we see symptoms. So when we use the word disease, disease means symptoms. Infection is when the pathogen actually infects the plant. So we can have infection that occurs and then conditions not quite be optimal for disease development, and that disease will remain latent or dormant or quiescent, whichever word you want to use. So it can just hang out there for a very long time. So usually a tree, a nice vigorous tree, will ward off any disease, if you will. So infection can kind of be there. Then if a tree becomes stressed, something happens to the tree, you can see disease kind of take off and symptoms kind of spread really rapidly when in fact that pathogen has been there all the time. Now, when we have wounds, absolutely, maple are really famous for having that oozing canker, that bleeding canker in that thin bark. You'll see those kind of red spots in a maple trunk, and that is indeed Phytophthora that can come in with wounding from a borer or from us, right, from mechanical damage. So it is possible. Mostly unlikely, but it absolutely is possible. Anything's possible in the biological realm. Anything else? All right. So there is the link again for the quiz. This is quiz number 5. This is webinar number 5. We have 2 to go. The next one will be combination of viruses and nematodes. We're going to look at some ELISA testing. I know a lot of you see your reports and we see ELISA testing and you don't know what the heck ELISA is. We're going to look at how those antibodies are put into basically a dipstick test. Phytophthora and Pythium are tested with ELISA. I chose to delay that. We're going to look at it with viruses next week. Maybe something that you would use if you have a specific grower or something, typically something that would happen in the diagnostic lab. But I think it's relevant to cover. It's really relevant to cover. So viruses and nematodes next, and that is webinar number 6. That is, I think, on the 10th of August. It's early August. I will be out next week. No, not next week. I will be out the week after that for a conference. Next week, I will be around. If anybody has any questions with any of this, if any of you want samples, remember, if any of you don't have things like hand lenses and you need them before the practicum, let us know. We really want to help everybody start practicing these skills now. Also, I'm going to ask Kim to go back and put the videos back on the site. They are not, the quizzes will not be open, the surveys will not be open, but the video will be there if you need to backtrack for any information, especially if you have a situation in your county and you need that information. Anytime I'm here to help you by text message, by Facebook or Twitter, by preferably email, phone call, whichever you need, I am always here. So I want to help you practice these skills and be able to solve problems right there for your stakeholder and not have the delay with us. And finally, remember, if you have a commercial sample, you can play with it on your own, but please get a sample to the diagnostic lab for confirmation especially when a commercial grower is depending on an absolute definitive answer. So with that, everyone have a good weekend. I will stay on for just a sec, so anybody who has last minute questions, and the link we'll keep putting it up for just a few for anybody who wants to click it. So happy weekend. Everybody stay dry. It looks like we're going to get some rain, so maybe we'll have some Oomycete activity with this cool rainy weekend. So all right, goodbye everybody.